the next question. And I must say thanks to Ayanda for printing all of these out for me. What a star. So here we go. This is from, guys, with all your funny ways that you write your names on Facebook, Lumetu de North Prince, I think. So, Lumetu de North Prince. All right, here we go. The diagram below shows the structure of a Malpighian body. So I hope you all know that a Malpighian body consists of the Bowman's capsule and the glomerulus. Now, the way you remember the word glomerulus is when there is a conglomeration, it means there's lots. So it gloms together like a glob, glomerulus. Okay, and a Bowman's capsule, it bows in the middle. So that's easy. All right, so Malpighian body, study the diagram and answer the questions that follow. Okay, now let's quickly draw our Malpighian body. People, you've got the kidney, okay, and you've got the renal artery and the renal vein, and then you have the urethra. Is it the ureter or the urethra? Come, tell me, think. Is this the ureter or the urethra? And I want you all to say, I know what it is. I know what it is, because we did the kidney last week. I'm not going to draw all of the rest. So this is the cortex. This is the medulla. Okay. And for each one of these little lines that comes down here for the pyramid, okay, is a little thing that looks like this. It's got a little blumps, and then it goes wiggle, wiggle, down, up, wiggle, wiggle, down. Okay, and this little thing here, so that goes wiggle, wiggle, down, up, wiggle, wiggle, down. And then you've got a whole bunch of these attaching into one tube. This thing here is called a nephron. And a nephron is the filtering unit of the, what's this thing? Kidney. Now, have you remembered what this pipe here is called? Come on. The urine's going to come here, and the urine collects here, and then the urine goes into the bladder. What is this called? It is the urethra or the ureter. Come on, people. Which one? Come, come, come. Urethra, it thrusts, or the ureter, it drips. And we did this last week. I can hear you all saying to me, Kathy, please, you know it is the what? It is the ureter, correct. And coming from the bladder, we are going to then have here, after the bladder, coming out of the bladder, that is where we're going to have the urethra. All right, cool. Now, we have to look at the Malpighian body. Now, remember, the nephron's job is ultrafiltration. Okay, so that is for ultrafiltration. It is for ultra reabsorption. Why reabsorption? Because you've already absorbed everything in the digestive system. So you are now going to reabsorb it. Why? Because guess what? We need to filter it. So the nephron's job, ultrafiltration, ultra reabsorption, and tubular secretions. Okay, now ultrafiltration happens here. Okay, then we have ultra reabsorption takes place there. And when we get to this part and that part, we have, oh, and a bit here, we have tubular secretions. So that is why this little thing is so important. And there are thousands of little nephrons everywhere with the Malpighian body sitting in the cortex and the tubules sitting in the medulla. Alrighty, now we're going to do our question. So let's get more space here. So we're going to do our thing. So here we go. We start with the Bowman's capsule. Okay. And now coming in here, we have a blood vessel, and it then goes, this is thin, and this is wide. And the way I've done it now, I look like an idiot. Let's just quickly do this section again. It's not easy drawing with this stupid pen. Okay. Okay, now, and then here we have a whole bunch of little capillaries. Okay, now, this thing here is C. This is D, so they want this lining here. Then this is E, this is B, and this is A. Blood comes in there, blood leaves there, okay? And something else is the blood that goes in here and the blood that comes out there, there is no gases lost here. Okay, which means that this is still an arteriole, and this is also an, let me just get this off here. This is also an arteriole. Okay, now remember, we go from the heart. When it moves away from, when a vessel moves away from the heart, okay, if it moves away, it is carrying oxygenated blood, number one, and it is carrying nutrients to give to the rest of the body. So when it moves away, it is an artery. A for away, A for artery, and your main artery that moves away from the heart is the aorta. So aorta, away, artery. Anything that moves is on its way back to the heart. So the inferior venae cavae, the superior venae cavae, if it's going back to the heart, it is a vein. That is also why when next year, when you do human reproduction and you have a little fetus and it's being supplied with blood from the placenta, okay, the blood coming from the fetus is going away from its heart, so therefore that is an artery, even though it's carrying deoxygenated blood. And going into the little embryo or the fetus is going, or the fetus, the embryo, going to the baby, the blood going in is coming to the heart, therefore it is a vein. So that is why it is switched over from the mother's blood supply. Just remember that. Your lungs, the same thing. Your pulmonary vein, at least your pulmonary artery, moves away from the heart. Arteries move away from the heart. And where does it take the blood? To the lungs. So your pulmonary artery is carrying deoxygenated blood, and your pulmonary vein comes from the lungs back to the heart. That's why it's switched around. All right, now let's get this. Okay, so we have this one here is large, or it's larger than this one, which is smaller. Okay, so this arteriole is larger, this one is smaller. That's something you have to remember here, very important. Okay, this thing here is a whole bunch of capillaries sitting here. And then you have this cavity, Okay, and then it goes down. Now remember, we're looking at that. We're just looking at this piece here. All right, now let's do our labels. A is going to be your afferent arterial. Afferent because it is arriving. And that's how you're going to remember it. Afferent arterial, and it is arriving with blood coming in here. This one, this B, is your efferent arterial. Efferent, remember, exit. So your afferent arterial arrives at the Malpighian body, and your efferent arterial exits the Malpighian body. All right, now, so that's A is afferent, B is efferent arterial. Why is it still an arterial? Because it is still going through the kidney. It is not on its way back to the heart yet. Okay, so it's still an arterial. Remember, we go artery, an artery split up into arterioles, and arterioles split up into a capillary network. Okay, and they will join up to venules, and venules all join up to veins. Okay, people, you must remember that. Okay, in fact, this is very important for you to understand. Alrighty, now I've moved up too high. Okay, AB. C is the glomerulus. Okay, it's just a glob. So think of glob, spell it. Break it up. Glomerulus. Then D, now that is, it can be the wall of the Bowman's capsule, or, see, I don't know what your teachers are teaching you, or it can be the squamous. I'm going to just rub this out so I've got space here. Oh man. Okay. So this is either the wall of the Bowman's capsule, or it can be the squamous epithelium, which is what this wall of the Bowman's capsule is made of, with very special little structures called podocytes. So I'm not sure what the examiners would have wanted there, but both of those are correct. Then for E, well, this is simply the cavity, or I don't know what we can call it, the cavity of the Bowman's capsule. Okay, but E I would have put as the Bowman's capsule. All right, so what happens now? Let's look at our questions. It says label parts A, B, C, D. So we've got A, afferent arterial, B, efferent arterial, C is the glomerulus, D is either the wall of the Bowman's capsule, and then you can say which contains squamous epithelial with podocytes, and then you cover both bases, and E is the Bowman's capsule. All right. Now, name and explain the process that occurs in the Malpighian body. All right, so let's name and explain that process. Let's use yellow. Okay, it's called ultrafiltration. Remember I told you right in the beginning, we have ultrafiltration that happens in the Bowman's capsule. And that is very, very, very important. All right, so how does this work? It starts with the afferent arteriole is larger than the efferent arteriole. Okay, and what does this do? This creates pressure as the blood flows through the glomerulus. All right, so let's just look here. Let me explain it. Okay, we've got the blood coming in here. Okay, and here the blood goes and it's wide. So this is large, it's bigger. And this end is smaller. Now, if you take a hose pipe, okay, and you are busy watering the garden, and what do you do if you want the water to spray further? You put your finger over the nozzle, all right? And if you put your finger over the nozzle, what are you doing? You're making it smaller. The minute you make it smaller, it creates pressure, okay? And you can try this with any tube that you connect to a tap at home. If you connect a tube to a tap at home, all right, the minute you make the front end smaller, it creates pressure always. So afferent arteriole is larger than the efferent arteriole, and that creates pressure in this whole bunch here. This whole bunch of capillaries here that make the glomerulus. That's where we have the pressure. All right. Now, what does that pressure do? The pressure, what does that pressure do? It makes sure that all the stuff in the blood here passes through into the Bowman's capsule cavity. Okay, so it pushes it through into here. And that is part of ultrafiltration. Now, we haven't finished. Then, we have the afferent is larger than the efferent, and this creates pressure. That's important. That's number one. Number two, our next point is that the inner wall of the glomerulus has podocytes which create slit pores for ultrafiltration. Okay, now let me explain this. You've got this single layer of squamous epithelium, hey, inside the Bowman's capsule. So let's draw it. We have this, here's the that's your inside part of the Bowman's capsule. Okay, here we have a single layer of cells all the way along here. And those cells are the same cells that you have in your mouth. Okay? Can you remember from grade 10 what cells line your mouth? They are called squamous epithelium. Okay, squamous because they're actually squashed. They actually look like this. Okay, now, sitting overlapping those squamous epithelium, girls, you all have those clips. You know when you take your hair up and you clip it? Right, those clips look, it's called a butterfly clip. It does this. That's exactly what the podocytes do around the capillaries. So the podocytes sit between the squamous epithelium and the capillary, which is here from the glomerulus. There's your capillary. Okay? And remember, your capillaries have a single layer of endothelium. Okay, which means things can move in and out very quickly in those capillaries. All right, now we've got the podocytes, and the podocytes sit here like this. And then we have another one sitting here. And where the podocytes do this, you have your slit pores, which are tiny. And those tiny little slit pores are tiny little gaps for these things, all these substances, to move into the Bowman's capsule. Okay, so the inner wall of the glomerulus has podocytes, which create slit pores for ultrafiltration. Now, okay, this means that glucose, amino acids, do you remember that amino acids are your monomers of proteins? Glucose, amino acids, mineral salts, vitamins, you see, when I'm vitamins, goodness, Kathy. Okay, vitamins, then your waste products, urea, uric acid, ammonia, what do you get when you're creatinine, when your muscles break down? Creatinine. You also have water. Okay, all of these are filtered and move into the Bowman's capsule. Okay, and now this is where the important thing comes. Because of those podocytes and the slit pores and the micropores that are in the capillaries, what do we have? We have the bigger molecules are left behind. So let's just zip this. Where's my eraser? Dump. Gone. Okay. But the podocytes and micropores do not allow red blood corpuscles or red blood cells, proteins, fats, which are large, and any other large molecules to pass into the Bowman's capsule. Okay, people, that is ultrafiltration. So let's just quickly recap. You have the endothelium of the glomerulus, which is a capillary. You have the inner layer of the Bowman's capsule, which is squamous epithelium, and a whole bunch of podocytes. So we've got slit pores, we have micropores, and they stop. They keep your red blood cells, they keep your proteins, they keep your fats in the blood. They will carry on through the efferent arteriole. Okay, on their way back to do their thing. Right, the glucose, the small molecules, glucose, fructose, galactose, amino acids, mineral salts, vitamins, water, the bad things, the urea, uric acid, creatinine, all of that passes through into the Bowman's capsule. Now, that's where the selective reabsorption comes in. Because when you go into the proximal and the loop of Henle and the distal, what happens? You've got to get all those things back. And that is when you see, for example, sugar or glucose in a person's urine. That's because there is so much glucose that that poor little nephron, which would normally be able to take all the glucose out and back and reabsorb it back into the blood. But if you have diabetes, your insulin isn't working, and you are not storing your glucose. So there's a lot of glucose in the blood. And a lot of glucose will then go into the kidney. And the little nephron cannot reabsorb all the glucose. And that's why you end up with glucose in your urine. But you know something? You will never see proteins in your urine. Ever. And the only time you will see red blood cells or blood in your urine is if something has broken. In other words, there's been a rupture. Okay, why? Your red blood cells need to transport oxygen. You don't want them messing around and having to be reabsorbed in your kidneys. Also, you've got the structures of the tubules aren't designed, they're designed for ultra reabsorption. They can't reabsorb large molecules, which means you would just wee out all your red blood cells and all your protein. You don't want to. You want to keep that in your body. Okay, so that is why it is ultrafiltration and ultra reabsorption. And your large molecules pass out to the efferent arteriole. And if I was examining you, that is what I would ask you. Not just, what's this, what's that? I would want to know what you would have going into the glomerulus and what you would have leaving the glomerulus, and then what the different regions of the nephron are designed for. That is structure versus function. People, you must learn structure versus function. And not just you, the matrics as well, the grade 10s as well. Always learn, if a thing looks like this, what is its job? Because let me tell you something now. The only useless thing you have in your body, and we all have something useless in our body. Do you know what it is? Have any of you got any idea? It's the appendix. Okay, it's just this useless piece of nothing, that tissue that we used to use apparently before when we were still eating lots of seeds and hard things. Okay, but at the end of the day now, it is just a nothing. Okay, that just causes issues. All right, now, next question. Last question, I think. Name the specialized cells found at D. Well, we've already spoken about them. The specialized cells here would be the podocytes. And then describe the significance of the shape of the structure at E, which means the Bowman's capsule. Now, people, look at the Bowman's capsule. It's a round, bowl-like structure. So what do we have? We have, instead of this flat thing, we've got this. We have increased the surface, and sitting, nestling inside that bowl of Bowman's capsule, you have the glomerulus. So there's a lot of area that is in contact between the glomerulus here, okay? Let me get another color. Between the glomerulus here and the Bowman's capsule over there, there's a lot of area, you see? So the shape of the Bowman's capsule allows the glomerulus capillary network to go into the bowl, and it increases the surface area for ultrafiltration, okay, which is really, really, really important. All right, so there you have it. Done, done, done. So grade 11s, that means it is the end of your session. And now grade 12s, get ready for genetics.